Hi, you are most welcome to Study Point. Today I give you all the question with answer of the chapter 2 of second year biology thought this video, so don't skip. See to the end. Question 1. Name the parts of an angiosperm flower in which development of male and female gametophyte take place. Answer 1. The male gametophyte or the pollen grain develops inside the pollen chamber of the anther, whereas the female gametophyte, also known as the embryo sac, develops inside the nucellus of the ovule from the functional megaspore. Question 2. Differentiate between microsporogenesis and megasporogenesis. Which type of cell division occurs during these events? Name the structures formed at the end of these two events. Answer 2. The difference between microsporogenesis and megasporogenesis is given below. Both events, microsporogenesis and megasporogenesis, involve the process of meiosis or reduction, division, which results in the formation of haploid gametes from the microspore and megaspore mother cells. Microsporogenesis results in the formation of haploid microspores from a diploid microspore mother cell. On the other hand, megasporogenesis results in the formation of haploid megaspores from a diploid megaspore mother cell. Question 3. Arrange the following terms in the correct developmental sequence. Pollen grain, sporogenous tissue, microspore, tetrad, pollen mother cell, male gametes. Answer 3. The correct development sequence is as follows: sporogenous tissue, pollen mother cell, microspore, tetrad, pollen grain, male gamete. During the development of microsporangium, each cell of the sporogenous tissue acts as a pollen mother cell and gives rise to a microspore tetrad containing 4 haploid microspores by the process of meiosis, microsporogenesis. As the anther matures, these microspores dissociate and develop into pollen grains. The pollen grains mature and give rise to male gametes. Question 4. With a neat labeled diagram, describe the parts of a typical angiosperm ovule. Answer 4. An ovule is a female megasporangium where the formation of megaspores takes place. The various parts of an ovule are: funiculus. It is a stalk-like structure which represents the point of attachment of the ovule to the placenta of the ovary. Hilum. It is the point where the body of the ovule is attached to the funiculus. Integuments. They are the outer layers surrounding the ovule that provide protection to the developing embryo. Micropyle. It is a narrow pore formed by the projection of integuments. It marks the point where the pollen tube enters the ovule at the time of fertilization. Nucellus. It is a mass of the parenchymatous tissue surrounded by the integuments from the outside. The nucellus provides nutrition to the developing embryo. The embryo sac is located inside the nucellus. Chalazole. It is the based swollen part of the nucellus from where the integuments originate. Question 5. What is meant by monosporic development of female gametophyte? Answer 5. The female gametophyte or the embryo or sac develops from a single functional megaspore. This is known as monosporic development of the female gametophyte. In most flowering plants, a single megaspore mother cell present at the micropylar pole of the nucellus region of the ovule undergoes meiosis to produce 4 haploid megaspores. Later, out of these 4 megaspores, only one functional megaspore develops into the female gametophyte, while the remaining 3 degenerate. Question 6. With a neat diagram, explain the 7-celled, 8-nucleate nature of the female gametophyte. Answer 6. The female gametophyte embryo sac develops from a single functional megaspore. This megaspore undergoes 3 successive mitotic divisions to form 8-nucleate embryo sacs. The first mitotic division in the megaspore forms 2 nuclei. One nucleus moves towards the micropylar end while the other nucleus moves towards the chalazal end. Then these nuclei divide at their respective ends and redivide to form 8 nucleate stages. As a result, there are 4 nuclei each at both the ends, i.e. at the micropylar and the chalazal end in the embryo sac. At the micropylar end, out of the 4 nuclei only 3 differentiate into 2 synergids and 1 egg cell. Together they are known as the egg apparatus. Similarly, at the chalazal end, 3 out of 4 nuclei. Differentiates as antipodal cells. The remaining 2 cells of the micropylar and the chalazal end move towards the center and are known as the polar nuclei, which are situated in a large central cell. Hence, at maturity, the female gametophyte appears as a 7-cell structure, though it has 8 nucleate. Question 7. What are gasmogamous flowers? Can cross-pollination occur in clistogamous flowers? Give reasons for your answer. Answer 7. There are 2 types of flowers present in plants, namely oxalar and viola minus gasmogamous and clistogamous flowers. Gasmogamous flowers have exposed anthers and stigmata similar to the flowers of other species. Cross-pollination cannot occur in clistogamous flowers. This is because clistogamous flowers never open at all. Also, the anther and the stigma lie close to each other in these flowers. Hence, only self-pollination is possible in these flowers. Question 8. Mention 2 strategies evolved to prevent self-pollination in flowers. Answer 8. Self-pollination involves the transfer of pollen from the stamen to the pistil of the same flower. Two strategies that have evolved to prevent self-pollination in flowers are as follows. In certain plants, the stigma of the flower has the capability to prevent the germination of pollen grains and hence prevent the growth of the pollen tube. It is a genetic mechanism to prevent self-pollination called self-incompatibility. Incompatibility may be between individuals of the same species or between individuals of different species. Thus, incompatibility prevents breeding. In some plants, the gynoecium matures before the androecium or vice versa. This phenomenon is known as protogeny or protandry respectively. This prevents the pollen from coming in contact with the stigma of the same flower. Question 9. What is self-incompatibility? Why does self-pollination not seed formation in self-incompatible species? Answer 9. Self-incompatibility is a genetic mechanism in angiosperms that prevents pollination. It develops genetic incompatibility between individuals of the same species or between individuals of different species. The plants which exhibit this phenomenon have the ability to prevent germination of pollen grains, and thus prevent the growth of the pollen tube on the stigma of the flower. This prevents the fusion of the gametes along with the development of the embryo. As a result, no seed formation takes place. Question 10. What is bagging technique? How is it useful in a plant-to-breeding program? Answer 10. Various artificial hybridization techniques under various crop improvement programs involve the removal of the anther from bisexual flowers without affecting the female reproductive part pistil through the process of emasculation. Then, these emasculated flowers are wrapped in bags to prevent pollination by unwanted pollen grains. This process is called bagging. This technique is an important part of the plant-to-breeding program as it ensures that pollen grains of only desirable plants are used for fertilization of the stigma to develop the desired plant variety. Question 11. What is triple fusion? Where and how does it take place? Name the nuclei involved in triple fusion. Answer 11. Triple fusion is the fusion of the male gamete with two polar nuclei inside the embryo sac of the angiosperm. This process of fusion takes place inside the embryo sac. When pollen grains fall on the stigma, they germinate and give rise to the pollen tube that passes through the style and enters into the ovule. After this, the pollen tube enters one of syzygids and releases two male gametes there. Out of the two male gametes, one gamete fuses with the nucleus of the egg cell and forms the zygote syngamy. The other male gamete fuses with the two polar nuclei present in the central cell to form a triploid primary endosperm nucleus. Since this process involves the fusion of three haploid nuclei, it is known as triple fusion. It results in the formation of the endosperm. One male gamete nucleus and two polar nuclei are involved in this process. Question 12. Why do you think the zygote is dormant for some time in a fertilized ovule? Answer 12. The zygote is formed by the fusion of the male gamete with the nucleus of the egg cell. The zygote remains dormant for some time and waits for the endosperm to form, which develops from the primary endosperm cell resulting from triple fusion. The endosperm provides food for the growing embryo and after the formation of the endosperm, further development of the embryo from the zygote starts. Question 13. Differentiate between A. Hypocotyl and epicotyl, B. Coleoptile and coleorhiza, C. Integument and testa, D. Perisperm and pericarp. Answer 13. A. The difference between hypocotyl and epicotyl is given below. Answer 13. B. The difference between coleoptile and coleorhiza is given below. Answer 13. C. The difference between integument and testa is given below. Answer 13. D. The difference between perisperm and pericarp is given below. Question 14. Why is apple called a false fruit? Which parts of the flower forms the fruit? Answer 14. Fruits derived from the ovary and other accessory floral parts are called false fruits. On the contrary, true fruits are those fruits which develop from the ovary, but do not consist of the thalamus or any other floral part. In an apple, the fleshy receptacle forms the main edible part. Hence, it is a false fruit. Question 15. What is meant by emasculation? When and why does a plant breed employ this technique? Answer 15. Emasculation is the process of removing anthers from bisexual flowers without affecting the female reproductive part pistil, which is used in various plant hybridization techniques. Emasculation is performed by plant breeders in bisexual flowers to obtain the desired variety of a plant by crossing a particular plant with the desired pollen grain. To remove the anthers, the flowers are covered with a bag before they open. This ensures that the flower is pollinated by pollen grains obtained from desirable varieties only. Later, the mature viable and stored pollen grains are dusted on the bagged stigma by breeders to allow artificial pollination to take place and obtain the desired plant variety. Question 16. If one can induce parthenocopy through the application of growth substances, which fruits would you select to induce parthenocopy and why? Answer 16. Parthenocopy is the process of developing fruits without involving the process of fertilization or seed formation. Therefore, the seedless varieties of economically important fruits such as orange, lemon, watermelon etc. are produced using this technique. This technique involves inducing fruit formation by the application of plant growth hormones such as auxins. Question 17. Explain the role of tapetum in the formation pollen grain wall. Answer 17. Tapetum is the innermost layer of the microsporangium. It provides nourishment to the developing pollen grains. During microsporogenesis, the cells of tapetum produce various enzymes, hormones, amino acids, and other nutritious material required for the development of pollen grains. It also produces the exine layer of the pollen grains, which is composed of the sporulinan. Question 18. What is apomixis and what is its importance? Answer 18. Apomixis is the mechanism of seed production without involving the process of meiosis in syngamy. It plays an important role in hybrid seed production. The method of producing hybrid seeds by cultivation is very expensive for farmers. Also, by sowing hybrid seeds, it is difficult to maintain hybrid characters as characters segregate during meiosis. Apomixis prevents the loss of specific characters in the hybrid. Also, it is a cost-effective method for producing seeds. At last, I want to say that if this video is helpful for you, then subscribe the channel and also click on the bell icon for latest updates.